I'm very pleased to share this session and to introduce my co-chair, Professor Peter Steinmann. He's professor at the University of Basel and deputy head of the Health System Support Unit at the Swiss Tropical and Public Health Institute in Switzerland. And I am especially pleased to introduce the first presenter, and it's Dr. Bouke de Jong, who is the head of the Mycobacteriology unit at the Institute of Tropical Disease, and who is my successor. So please, Bouke, you have the floor for 8 minutes.
It's an honor and a pleasure to present to you on behalf of a large team the studies that are running in the Comoros. And I will focus on the laboratory results and specifically on drug resistance surveillance, which is all the more important when we are administering prophylaxis for leprosy with rifampicin. So the first study that I'll present is the ComLep study. And this afternoon, you will hear from Professor Hasker a more detailed update on the people study. And this is a timeline. So the ComLep study ran from 2017 to 2019, and we recruited all leprosy patients in the island of Anjouan. And since 2018, we started also the people cohort study, which includes a cluster randomized trial of prophylaxis with rifampicin. But in addition, we continue to recruit patients with leprosy disease. And in the present analysis, I'm sharing with you the results of patients recruited from 2017 to 2020. So as you know, the Comoros are an island group in between Madagascar and Mozambique. And of the 3 islands, the 2 islands of Moélie and Anjouan are leprosy endemic. So on Anjouan, where the ComLep study took place, population is around 340,000 inhabitants, with a leprosy incidence around 7 per 10,000. Since 1980, multi-drug therapy has been administered, and since 2008, active case finding, first in the form of skin camps, has been introduced. So despite really active and excellent leprosy control program, the incidence remains high, but the number of patients, the proportion with grade 2 or greater disability at time of diagnosis of leprosy is fortunately low. And the WHO classification for the diagnosis is used based on the number of lesions as multibacillary or possibacillary. So patients recruited into the study are asked for informed consent to take a biopsy, a nasal swab, finger stick blood, and slit skin smear only of the multibacillary patients. And then we proceed with molecular diagnostics to determine the bacillary load. And if this is sufficiently high, also with genotyping. So I'm going to share some results of the skin biopsy. The target is used RLep by qPCR detection. We're using the primers as designed by Dr. Truman's team. And as you can see, for possibacillary patients, 68% tested qPCR positive for RLep. And in these studies, we took negative controls along all the way from sampling through DNA extraction and through the molecular analyses. And these were always negative as they should be. In the multibacillary patients, 80% test positive for the RLep qPCR. If we then go to the nasal swab, the positivity is much lower. Only 3% of possibacillary patients tested RLep positive. And 25% of multibacillary patients. And these were indeed the patients who also had a higher bacillary load on their skin biopsy than the ones who tested negative by nasal swab. The finger stick blood is tested for antibodies to PGL1. And in possibacillary patients, 53% tested PGL1 positive, whereas for multibacillary patients, 73% tested NTP-GL1 positive. And then for the slit skin smears, only on patients with clinically multibacillary disease. And the slit skin smears were taken from the earlobes and the lesion. 45% tested microscopy positive. So genotyping, the biopsies were split in two. For the one hand, for DNA extraction followed by duplex MICLEP, which I'll explain in a minute. And this is a target deep sequencing assay where specific targets of interest for drug resistance or molecular epidemiology are amplified and subsequently genotyped with an Illumina platform. Whereas for whole genome sequencing, which Sophie Braat is currently learning from Charlotte Avanzi, the entire genome of M. leprae is amplified and sequenced. So the duplex MICLEP is a very novel assay developed by Philip Suffies, who identified the targets, and developed by a GenoScreen Philip Suppliant team. And we have validated its use against the classical techniques for drug resistance typing and genotyping. And as we discussed, so there's drug resistance related genes involved, but also VNTRs and SNP types for molecular epidemiology and to study chains of transmission. And then after a multiplex PCR, there is next generation done on the Illumina platform. And right now I'll focus on the 6 drug resistance related genes. So these are the drug resistance related genes involved for resistance to dapsone, fluoroquinolones, and rifampicin. And the ones with a thick line are recommended by WHO for the performance of drug resistance surveillance for leprosy. And in addition, we included a few more targets for rifampicin and also a gene that may confer hypermutability. In this test, where we first do a PCR and then deep sequencing, we are also able to detect minority populations. So resistance doesn't happen overnight where all the bacilli are resistant, but it starts with a small population. Let's say 10% here, the red ones are resistant to rifampicin. If a patient then continues to receive rifampicin-based treatment, then this population will be selected for. So then it will become 40% and ultimately it will be a fixed mutation at 100%. So in order to detect early signs of acquired resistance, it's important to have a test that already can recognize small proportions of mutants. So here's the study team. It's really an incredible team led in the middle by Dr. Yunusa Assoumani, clinical epidemiologist. In 2017 to 2020, we identified almost 1,200 patients with leprosy, of whom 86% agreed to be sampled. And also, if they only had a lesion on the face, we do not take a biopsy. From these patients who were sampled, you can see the breakdown into those with high bacillary load and lower. And we ultimately included 260 DNA extracts from biopsy for the duplex MICLEP target deep sequencing. So this is a slightly complex figure, but on the top you see that on the left, some patients had already started treatment at the time of sampling and some hadn't. And on the top, you can see that some patients came from villages where in 2015 already single dose rifampicin post-exposure prophylaxis had been administered. And one of these patients had indeed received SDR-PEP and then subsequently developed leprosy and was included in the study. And then in the middle, you see those villages where single double dose rifampicin post-exposure prophylaxis was distributed in 2019 as part of the PEOPLE study, which will be presented by Dr. Hasker. And also from these villages, one patient had received post-exposure prophylaxis and subsequently developed leprosy. And then in the bottom, you see that 9 patients had previously been treated for leprosy and were included in this analysis. So the results of the 260 patients included most of the targets were amplified successfully, and none of the rifampicin-related or dapsone-related genes showed any mutations. For the fluoroquinolone-related genes, we identified 7 isolates, 7 samples with a mutation, the same mutation in gyrase B. But these patients were both clustered by the rest of their genotype in diplex MICLEP and also from 2 adjacent villages. And we think that this is a phylogenetic mutation that is not related to fluoroquinolone resistance. We also included a protein structure analysis, which again is suggestive that is not a fluoroquinolone conferring mutation, although to be sure you would have to test this in mouse footbed analysis with drug exposure. So in summary, we did not identify any drug resistance, not even minority population, for any of these drug resistance related targets in the leprosy patients in the Comoros. And moreover, we now believe we have a really useful tool for the early warning for drug resistance emergence in the context of post-exposure prophylaxis. And this can also be applied on biopsies stored in ethanol and in remote areas where molecular platforms are not available. So I thank you very much and give credit to this incredible team from the Damien Foundation in the Comoros who served the patient with leprosy so well. Thank you very much.
Thank you, Bouke, for this excellent presentation. It's a good news if there is no resistance. Professor Steinmann, did you receive some questions?
Yes, thank you very much also, Bouke, for the very nice presentation, very informative. And indeed, we have one question from the Q&A chat. And if others from the audience have questions, it's not too late. Please type them there and we can read them out. So the first question we have received, Wim wanted to know, is a genetic determination of resistance also possible for clarithromycin?
Yes, I admit that I do not know which genes are included, and it's not part of the current version of the diplex MICLEP. But just like for the TB, where the diplex has already much more mileage, it is possible to add drug resistance targets. So for the TB one, there's now a XL version where also drug resistance to new TB drugs is included. So it's an excellent suggestion that if clarithromycin is used for leprosy post-exposure prophylaxis, to suggest to genoscreen to include genes involved in resistance to clarithromycin in the next version of the diplex MICLEP.
Very good, thank you. Then we have a second question, this time coming via the chat. The question from Diana is, why were slit skin smears only taken from the earlobe and not from the lesion?
Maybe the figure wasn't clear. So there are 3 spots on each slide, and 2 are from each earlobe and 1 from the lesion. And we only do this for multibacillary patients who are clinically with more than 5 lesions, because the team feels that in possibacillary disease, the slit skin smears are rarely positive, and it's still not a very comfortable test. So yes, the lesion is included. Sorry about that.
Very good, thank you for this clarification. And the last question from Diana. This is good news that rifampicin resistance has not yet appeared. However, we shall have to have monitoring over a long time to detect mutations occurring later. Do you want to comment on this? It's rather a comment than a question, but do you want to say anything to this?
I fully agree. And I think because we only were able at this point to include 2 patients who themselves developed leprosy after having received a post-exposure prophylaxis, it's too early to really exclude that such resistance may be induced. And also with leprosy having such a long incubation period, especially after rifampicin, we need to really have long-term follow-up and monitoring to make sure that we do not induce resistance. I see another in the chat. I can.
Yeah, in the chat appear more questions now. Do we still have time, Professor Françoise? What is?
You're muted, Françoise.
One more question.
One more question. Then the question which came first, is there resistance for MTB in the same area?
Also an excellent question. So in the Comoros, the gene expert MTB-RIF was introduced in 2017, and they also do excellent TB control. So there has not been a single case of rifampicin resistance since the expert was applied to all TB patients. So no RIF resistance has been induced by the use of leprosy PEP. And a very important question.
Very good. Thank you so much. And with this, I hand back from the questions to the next presentation.
Okay, the next presentation will be from Dr. Becky Rivoire from the American Leprosy Mission, and she will present some preliminary results with the vaccine LepVax. You have the floor for 8 minutes, please.
Thank you. American Leprosy Missions is committed to bringing an end to leprosy by developing an effective vaccine. In 2002, ALM partnered with Infectious Disease Research Institute, IDRI, to develop such a product. 20 years of research and $6 million has resulted in LepVax, a promising vaccine candidate for the treatment of leprosy, neuropathy, and prevention of disease. Results from the phase 1A trial has demonstrated vaccine safety and immunogenicity in healthy volunteers residing in the US, a non-endemic region for leprosy. Following a nearly 2-year interruption due to the COVID pandemic, LepVax clinical testing among people most at risk is now ready to proceed. LepVax consists of 2 active ingredients. The LepF1 antigen is a quadrivalent fusion protein engineered with 4 leprae T-cell antigens. And the adjuvant GLA-SE is a synthetic glycopyranosyl lipid A in a stable emulsion. It's a toll-like receptor 4 agonist that generates antigen-specific pro-inflammatory Th1 responses. Preclinical data support both a prophylactic and therapeutic vaccine application. The therapeutic potential was observed in armadillos pre-exposed to leprae, followed by inoculation with either saline, BCG vaccine, or LepVax. The proportion of animals presenting with normal, borderline, or abnormal nerve function are shown here in white, yellow, and red bars, respectively. LepVax results were significant compared to control and BCG groups with a p-value less than 0.05. Moreover, the prophylactic potential was observed in mice vaccinated with either adjuvant alone or LepVax. 1 month after the final immunization, animals were infected in the footpad with leprae, and 6 months after infection, footpads were harvested. The drop in bacterial burden was highly significant, with a p-value less than 0.01. These and other supporting in vitro and in vivo assays have led the team to develop a parallel clinical development pathway for evaluating LepVax for the treatment and the prevention of disease. The phase 1B trial will test safety and immunogenicity in healthy subjects and thereby serves as a bridging study for both indications. Our immediate focus is on treatment because of the nearly 4 million people post-MDT living with persistent consequences of leprosy disease. The phase 2A trial will test safety and immunogenicity in possibacillary leprosy patients with 0 bacillary index and mild disease before moving to test bacillary leprosy patients with more pronounced disease and greater risk of neurological damage. To implement this study, ALM serves as the funder, and IDRI is the owner of the technology and supplier of clinical material. Fiocruz is the clinical trial sponsor and where the study site is located. Further support comes from the Vaccine Advisory Group for LepVax, established to guide and counsel and direct major vaccine decisions. Voting members include Drs. Ann Ginsberg, Christika Singh, Warwick Britton, and Sinacio Talhari. Study objectives for the phase 1B and 2A trials are safety and tolerability of a low dose and high dose in healthy individuals and highest tolerated dose in PB leprosy patients. Cytokines, interferon gamma, IL-2, IL-4, IL-5, and TNF-alpha, as well as IgG antibodies against the LepF1 antigen will be measured to assess immunogenicity. The expectation is that LepVax will be safe and tolerable and immunogenic in healthy individuals and PB patients. A major milestone was accomplished with the submission of the drug dossier to the Brazilian Regulatory Authority Anvisa, followed by receipt of a list of their questions. As a result, improvements were made to the clinical protocol and development plan, and a detailed response facilitated by Fiocruz was sent back to the Anvisa. Document modifications primarily included the change of subject allocation ratios and increasing subject enrollment for statistical justification and loss to follow-up. Likewise, an introductory vaccine advisory group meeting was held with a follow-up meeting planned after the phase 1 study begins. Notwithstanding, Dr. David Block at the Erasmus University in the Netherlands was recruited to design 2 epidemiological models, like the ComLep models, to predict the impact of LepVax on leprosy burden, i.e. disability, stigma, etc., and leprosy transmission. Moving forward, we expect to receive Anvisa authorization to import clinical materials from IDRI by the end of May. Enrollment of healthy subjects in the phase 1B trial is expected in June. Meanwhile, the model for transmission is expected to be completed by the end of this year for generating predictions of the impact of LepVax on the lives of leprosy patients and prevalence of disease over time. I want to thank our members who have financially supported this project and the many researchers and advisors who have generously given of their expertise. Many of us know that if clinical research requires patience, intelligence, and dedication. And oh, did I mention money? This phase 1B/2A trial is the first key to unlocking the possibilities of LepVax for stopping and preventing the scourge of leprosy. Thank you for your time.
Thank you for this very clear presentation. Peter, did you receive some questions?
Yes, we have questions coming in. And also from my side, this is really exciting to see a vaccine moving towards the clinical trial phases. So the first question was, what is the absolute number of armadillos studied? Annemieke asked.
Yes, hello, Annemieke. There were 2 armadillo studies done with 6 to 8 animals in each of the studies per group. Treatments and control and BCG groups.
Very good. And then we have from, again, Annemieke, the second question. If anybody else also wants questions, please put them into the Q&A. What is the immune profile when only adjuvant is used? Is this tested as well in the trial in Brazil as to prevent the situation in with vaccines again in South Africa with vaccine against TB?
That's a good question. Right now, we have 2 treatment arms that'll be tested in each of these trials. One is a vaccine that contains the adjuvant and the LepF1 antigen, and the other arm is a placebo with just saline. So we will not be investigating adjuvant alone at this point.
Thank you. And until we have more questions, Diana asked, why is PB cases, if MB, are more at risk for neuropathy?
Thank you, Deanna. It's a good question. We are a little bit nervous about the Th1 TLR4 agonist application of the adjuvant. And so we're starting with PB leprosy patients that have a lower risk of developing further difficulties. And although we didn't see these in the armadillos, we're not expecting them, but it's a precautionary step approach.
Thank you. And I think this also answers the second question in the chat, which was also about the choice of PB patients. Last question, as far as I see, again, anemic, will antibody assessment be taken into account in immune monitoring in the Brazilian trial?
Yes, the antibody testing will include all forms of IgG, the IgG1, 2A, 2B, and 2B, 3, and 4. And those will be taken into account for both the phase 1B trial and 2A trial at Fiocruz.
Thank you so much. Bouke, you have a question I see live.
Yeah, I thought I would just take the opportunity to ask like this, and congratulations on the really important progress and crossing fingers for the trials. For your phase 3 trial, non-inferiority against what? Against post-exposure prophylaxis, or how will you measure the impact of the vaccine?
For the treatment of disease aspect?
I wasn't sure if it was a treatment or prophylaxis, but what will be your reference?
Yes. Right now, our reference point will be the, for the treatment of disease, our reference point will be neuropathy. We'll be measuring for neuropathy primarily, but we'll, of course, continue to evaluate the immunological aspects of the vaccine compared to the placebo.
Thank you so much. And with this, I hand back for our next presentation.
Okay, the next one will be Dr. Pushpendra Singh, who will speak about molecular mechanism of immunomodulation imparted to Mycobacterium indicus pranii vaccine against multibacillary leprosy. You have the floor for only 3 minutes.
Thank you very much, Professor Portaels. And with that, I would start this presentation about the Mycobacterium indicus pranii vaccine that has been approved for multibacillary leprosy in India as well as by FDA. So the leprosy patients are actually the major contributors in transmission and are also prone to reinfection because they do not mount any immunological memory against leprae. So they remain lepromin negative also, despite having taken the complete treatment also. In such cases, it has been reported that Mycobacterium indicus pranii vaccine can convert them into lepromin positive, and there have been several studies indicating better or faster bacterial clearance and histopathological improvement upon MIP plus chemotherapy administration. So Mycobacterium indicus pranii basically is a non-pathogenic mycobacterium, and its vaccine form actually involves autoclaved preparation. So there is also no concern about the its simultaneous administration along with rifampicin or any other anti-leprosy drugs. So in previous studies also it has been investigated for its mechanisms, but all such studies were carried out in a mixed group of cells, and there was no clear difference observed between vaccinated and unvaccinated arms when the composite population of the whole blood is used. So in this study, but there is clearly a phenotypic effect phenotypic difference between the bacterial clearance as well as in terms of the immunomodulation apparent from the cytokine and chemokine analysis. Therefore, in this study, we aim to perform immunoprofiling of MIP vaccinated and non-vaccinated individuals using flow cytometry. And during the flow cytometry, we will also separate different cell populations, and we will perform comparative blood transcriptome analysis on these separated population to understand differential gene expression in individual cells of group of cells instead of the mixture of different cell population. And in addition, the drug resistance related mutations and the genotypes would be investigated by pathogen genomics using sequencing approach. So there are the timelines are indicated here. Blood sample would be collected pre-vaccination from the patients who are being given vaccine and chemotherapy together, and after that the first dose of the vaccine will be administered. After that, after 1 month of the first dose of vaccination, there would be again a collection of blood, and a booster dose would be given after 6 months, and after that 1 month of the booster dose, there would be again a blood sample collection. So the experiment that we aim to perform are to using the 6 to 7 ml of the blood at these 3 time points. Those would be magnetically sorted to separate different cell population, and those would be stored in RNA stabilizing agent for RNA sequencing purpose, and the supernatant of the would be used for cytokine and chemokine analysis and ELISA based assays, and the cells would be used for immunophenotyping. So pathogen genomics work would be carried out to investigate whether there is a drug resistance related mutation in any of the arms, because those could be the confounding factor. And we aim to investigate 118 patients in each group. So far we have taken regulatory approvals for this study from the Institutional Ethical Committee as well as from the Scientific Advisory Committee meeting. And since this involves foreign funding, we had to get it approved from the Health Ministry's Screening Committee. And preliminary optimization experiments have been completed for the nucleic acid extraction and multiplex PCR, and for the fax-based analysis also, some preliminary experiments have been completed. And with that, I thank the LRI for supporting this project and the collaborators who are part of this study. Thank you very much.
Thank you. I have one question. Could you give us more information about this curious Mycobacterium MIP?
Yes. So,
where does it come from?
So this is actually soil Mycobacteria, and it is very closely, upon its whole genome sequencing, it closely clustered with the Mycobacterium avium group. It is not very close, but it has around 6 MB genome, and this has been actually described by, so earlier it was referred as Mycobacterium W, but there was a big confusion about the M. tuberculosis W lineage. That is why it was renamed based on its discoverer, Dr. From Talwar, Pran, and from NII. That is how it comes named as indicus pranii. But it's a non-pathogenic Mycobacterium.
Thank you.
Thank you.
So our last speaker.
Now, sorry, now suddenly the questions are coming. Do we have time or not?
Yes, yeah, one or two more questions.
Okay, then we, quick questions. Anouk asked, to which antigens do you want to measure the IgM and IgG response in the ELISA?
So against the whole cell lysate of leprae.
Thank you. And the second quick question, you are concerned about the LL patients, but most of them have done their transmission pre-diagnosis.
True, that is true actually. So it is actually more about the saving them from the reinfection and from stopping them from continuing the transmission. And
thank you
this
so much.
study,
And over to the next.
Thank you.
Okay, thank you. So the last speaker is Dr. Vino, and he speaks about the use of molecular methods in subclinical murine models of leprosy to examine the efficacy of proposed post-exposure prophylaxis regimens. And you have the floor for 3 minutes.
Thank you. So today, good morning to you all. Today I'll be presenting the use of molecular methods in subclinical murine models of leprosy to examine the efficacy of proposed post-exposure prophylaxis regimen. As we know that the multi-drug therapy is very effective, still there is subclinical infection among the leprosy patients, and that could be the major reason for continued transmission in highly endemic region. So there is a need for effective administration of post-exposure prophylaxis. And our major objective is to determine which rifampin-containing treatment is the most effective post-exposure prophylaxis. So here is our study model. We have chosen 3 strains of mouse, which has different immune status. In the graph, you can see that like biopsy mouse, which are resistant to M. leprae, and interferon gamma knockout, which is like partially susceptible to M. leprae, and you can see the athymic nude mice fully susceptible to M. leprae. So these are the graphs showing M. leprae growth in response to when grown in these strains. And here is the drug treatment plan. We are treated with single dose, so we will be treating with single dose of rifampin, double dose and 36 weeks after the treatment, we will be performing RLAP or RLPM, qPCR assay to determine the effect of drug on bacteria growth. So RLAP is the N-leprae specific repetitive sequence, and the RLPM is the N-lepromatosis specific repetitive sequence. Now the expected outcome or the impact of the study, and this study model will provide the most effective post-exposure prophylaxis regimen for contacts with different immune status. And this study model will provide information about which patients needs additional prophylactic treatment. And similar study design will be applicable to additional post-exposure prophylaxis regimens that may be proposed in the future. And finally, the clinicians can adopt optimum treatment plan when diagnostics become more sophisticated. So we would like to thank Leprosy Research Initiative and Turing Foundation for supporting the study. Thank you.
Thank you very much. And thanks to all the presenters, we will have some time for additional questions now. So I would like to thank you all for your excellent presentations. And the floor is now open for more questions if necessary. Peter, it's...
Yes, we have one. Thank you. Again, Annemiek asked, what is the readout of immunity after PEP in the immunocompetent mice?
Go back to the slide. Yeah, like the one is like a biopsy, which are resistant to M. leprae, and interferon gamma knockout, which is partially susceptible to M. leprae. The cell growth will be less, and athymic nude mice, as we know that M. leprae will be fully susceptible to M. leprae. So you can see the enhanced growth of M. leprae in athymic nude mice.
Okay, I think there is a follow-up question. What assays will you perform after PEP?
No, we'll be doing RLAP and RLPM, RLAP for the M. leprae and RLPM.
at least on my end, you are cutting out. Okay, let's move to the next question. There is one open one for Pushpendra, actually. Will you assess whether pranii induces trained immunity?
So basically, we would be looking at by the RNA sequencing only. So all the expressed genes that are differentially expressed between the vaccinated and non-vaccinated one, those would be revealed through comparative transcriptomics. And that would involve all the relevant genes actually.
Thank you. And with this, we have gone through all the questions which were arriving at my end. Anybody from the panel here, comments or questions? Yes, Bouke.
Annemieke asked a question on host genetic analyses for the Comoros. And I also want to apologize that I, in my excitement, didn't reach the very last slide with acknowledging all the funders and teams. But back to the host genetics, it's a very good point. The very first time I came to the Comoros in 2015, the local team said, we do everything that's recommended and we think we have high susceptibility in addition to malnutrition in our population. So the specific hypothesis was that Xeroderma pigmentosum, which is also it's a severe skin disease, which is common in the Comoros, that heterozygous individuals may be at higher risk of leprosy disease. So it's unlikely, actually. William Faber said that in Moshi in Tanzania, where there is a lot of Xeroderma pigmentosum, it's not shown to be associated with leprosy, but it highlights that genetic studies indeed would be a nice element to see if that explains the high incidence there. So I agree with Annemieke that it's a good point. And then I see another question.
Yes, exactly.
Can the D-plex also be used on slit skin smears? Yes, it can be. It all depends on the limit of detection. How much DNA do you have? So slit skin smears will provide less DNA than your average biopsy in volume of material from which you extract mycobacterial DNA. But of course, some of the multibacillary patients under the microscope have chock full of acid-fast bacilli, and then you'll have sufficient to also do the D-plex on the slit skin smear. So it depends on, it will be less successful on postbacillary patients or microscopy negative samples. Thank you.
Thank you so much. And with this, again, I think we have reached the end of the questions which have been submitted. So back to the moderator or chair, actually.
So no more questions? Well, we are great. It will be the coffee break at 3 o'clock. So goodbye. Thank you all for your excellent presentation. It's quite positive to see all these new development in the field of leprosy, vaccines, and prophylaxis, and so on. So thank you so much for this exciting session. And have a nice morning or afternoon or evening.
Thank you.
Thank you. Goodbye.